Mapping tissue ultrastructure is the direct route to clinical pathology. Every symptom begins as a structural failure at the microscopic level. This tri-split macrograph displays skeletal, cardiac, and smooth muscle. In panel A, skeletal fibers appear as massive, cylindrical cells with prominent cross striations. To maximize space for contractile proteins, their multiple nuclei are displaced to the extreme periphery, flattened directly against the sarcolemma. This arrangement allows the fiber to pack its cytoplasm with myofibrils, enabling the strong, quick, and strictly voluntary contractions required for skeletal movement. The highly ordered cellular structure of skeletal muscle is specialized for rapid, massive force generation. Panel B shows cardiac muscle. These striated cells are shorter and frequently branched, possessing one or two round, centrally located nuclei. The defining ultrastructural signature of cardiac tissue is the intercalated disc, appearing as dark transverse lines where myocardial cells join end to end. These discs facilitate mechanical and electrical continuity. Desmosomes provide the anchoring strength to prevent separation during contraction, while gap junctions allow rapid ionic flow necessary for synchronized pumping. Finally, panel C shows smooth muscle. This tissue consists of tightly packed, spindle-shaped cells that completely lack visible striations. Each cell contains a single central nucleus. During contraction, the shortening of the cell causes this nucleus to take on a characteristic corkscrew appearance. Smooth muscle is responsible for slow, involuntary movements. It provides sustained autonomic tone to regulate blood vessels and move contents through the digestive tract. Whether a muscle provides voluntary force, intrinsic rhythmic pumping, or sustained tone is determined by how its cells are fused and organized. Harnessing microscopic cellular contractions to move the macroscopic skeleton requires a nested hierarchy of connective tissue. The epimysium is the outermost layer, dense connective tissue enclosing the entire muscle. Septa extend inward as the perimysium, bundling fibers into fascicles and providing pathways for nerves and vessels. Within each fascicle, a delicate endomysium surrounds every individual muscle cell, containing a rich capillary network delivering oxygen directly to the fibers. This precise collagen scaffolding is what prevents the forces of contraction from tearing the muscle fibers apart, transmitting that mechanical energy directly to the tendons and bone. Deep within the cytoplasm, the sarcomere is the functional unit of striated muscle, stretching from one dense C disc to the next. The light-staining I band contains only thin actin filaments. The dark A band marks the entire length of thick myosin filaments. At its center, the lighter H zone contains only myosin rods bisected by the M line, which anchors them in place. During contraction, myosin heads pull actin filaments toward the center. They don't change length, simply sliding past one another, drawing Z discs closer together. As the sarcomere shortens, the I band and H zone narrow significantly. However, the A band remains at a constant width throughout the entire contraction. Millions of these microscopic sliding events occur simultaneously, generating the massive force seen at the macroscopic scale. Translating an electrical signal into a physical contraction requires T-tubules, deep invaginations of the sarcolemma that allow depolarization to reach the center of the fiber. These tubules interface directly with the calcium-storing sarcoplasmic reticulum. This electron micrograph shows the skeletal muscle triad. It consists of one central T-tubule flanked by two terminal cisternae of the sarcoplasmic reticulum. In skeletal muscle, these triads are located precisely at the A-band-I band junctions. Cardiac muscle uses a dyad instead, pairing one T-tubule with a single sarcoplasmic reticulum cisterna. These dyads are positioned at the Z discs rather than the AI junction. Smooth muscle lacks sarcomeres, T-tubules, and the regulatory protein troponin. Its contraction mechanism is fundamentally different. When calcium enters a smooth muscle cell, it binds to the protein calmodulin. This complex then activates an enzyme called myosin light chain kinase, or MLCK. MLCK phosphorylates the myosin heads, allowing them to bind with actin and initiate crossbridge formation. Smooth muscle filaments are anchored to dense bodies, which act as the functional analogs to Z discs. As the filaments slide, they pull on these dense bodies to squeeze the cell in a corkscrew fashion. The specific intracellular machinery, whether triads, dyads, or the calmodulin cascade, tunes the speed and rhythm of the contraction to the needs of the tissue. High structural specialization often limits the regenerative capacity of a tissue. Skeletal muscle has a limited repair mechanism that depends on satellite cells. These are progenitor cells that remain adjacent to mature fibers, between the sarcolemma and the external lamina, and proliferate to produce new fibers after an injury. Smooth muscle retains a much higher capacity for regeneration. It grows through both hypertrophy, an increase in cell size, and hyperplasia, the production of new cells via mitosis. Cardiac muscle is the most vulnerable, as it lacks satellite cells. Dead myocardial tissue cannot be replaced by new muscle. Instead, it is repaired through the formation of non-contractile fibrotic scar tissue. This permanent loss of functional architecture is the defining characteristic of progressive cardiac disease. Understanding these architectures is essential for interpreting the breakdowns seen in clinical pathology. In Duchenne muscular dystrophy, a mutation in the dystrophin protein disrupts the linkage between the internal actin cytoskeleton and the extracellular matrix. Without this anchor, the sarcolemma is easily damaged during contraction, leading to muscle fiber atrophy. Rigor mortis demonstrates a failure of the sliding filament mechanism. When ATP production stops after death, myosin heads cannot detach from actin, permanently locking the sarcomeres in a rigid state. Myasthenia gravis is an autoimmune condition where antibodies destroy acetylcholine receptors on the junctional folds of the sarcolemma. This cripples the neuromuscular junction, preventing the signal for contraction from reaching the myofibrils. In the case of a myocardial infarction, the destruction of cardiac cells causes unique sarcomere proteins, troponin I and troponin T, to leak into the blood. These serve as the gold standard biomarkers for diagnosing heart attacks. Clinical disease is a failure of architecture at the microscopic level. Mastering the foundational blocks of normal histology provides the necessary baseline for identifying where those systems break down.